Hello everybody, today we will be discussing proto-oncogenes and oncogenes. Specifically, what are they, what causes proto-oncogenes to become oncogenes, and how oncogenes cause cancer. So what is a proto-oncogene? They are normal genes within our body. They have an important role which includes regulating cell growth, regulating cell division, and controlling apoptosis. They are essential to keeping our body healthy. But what is the difference with an oncogene? There are mutated genes that have the potential to cause cancer. They are a result of mutated proto-oncogenes. The National Cancer Institute defines oncogenes as a gene that is a mutated form of a gene involved in the normal cell growth. Cancer can arise when a proto-oncogene is mutated, changing it into an oncogene and causing the cell to divide and multiply uncontrollably. Next, we look at what causes this mutation. Some examples are exposure to carcinogens or UV light. This damages DNA and alters proto-oncogenes with mutation, which becomes an oncogene. Some oncogenes work like an accelerator pedal in a car, pushing a cell to divide again and again. Others work like a faulty brake in a car parked on a hill, also causing the cell to divide unchecked. Next, how do oncogenes cause cancer? Normal genes which turn defective due to chromosomal rearrangements, mutations, or gene amplifications, leading the formation of oncogenes. The transformation usually occurs as a result of a mutation, often in the part of the gene responsible for controlling the gene's expression due to the environmental carcinogens and mistakes made by the cell's DNA replication machinery. Normally, the tumor suppressor gene wipes out any abnormal DNA quickly, therefore decreasing an oncogene's ability to make a normal cell cancerous. The product of oncogenes may lead to cell growth even when these other signals are not present. Lastly, let's look at the proto-oncogenes transformation phase. The first mode of activation is point mutations. Changes in a single base and insertion or deletion of DNA can result in the substitution of a single amino acid in a protein that changes the function. Second is the mode of activation is the gene amplification. Extra copies of the gene result in more of the gene product, proteins that lead to cell growth, being produced or expressed. And third mode of activation is the translocation and rearrangement. Movement of the portion of the DNA from one place to another can occur in a few ways. Sometimes a proto-oncogene is relocated to another site of a chromosome, and because of the location, there is a higher expression, larger amounts of protein produced. Other times, proto-oncogenes may become fused and another gene that makes the proto-oncogene, now an oncogene, is more active. And that is how a proto-oncogene becomes an oncogene. Thank you so much for watching.
Created using Powtoon.